Dr. Sen, and we can pass the ball now to Gary.
Thank you very much.
Okay, next I'd like to introduce Dr. Gary Van Berkel, who is the owner and CSO of Van Berkel Ventures, LLC, an analytical measurement, science, innovation, research, consulting, and writing firm in Oak Ridge, Tennessee. Prior to starting this company in the early 2018, Gary was a group leader and distinguished scientist in the mass spectrometry and laser spectroscopy group at the Oak Ridge National Laboratory. The title of his presentation is Nanoliter Droplet Dispensing for Analytical Mass Spectrometry and Other Applications. Please turn your attention to Dr. Van Berkel.
Shivo, who sponsored me putting together this presentation. So I've been very interested in droplet applications in mass spectrometry for a number of years, and that's what really led to me putting together this presentation. And I'm particularly interested in non-contact droplet dispensers, often called droplet on-demand dispensers, and their various uses in mass spectrometry. So you'll have the chance to go back and look at this presentation in detail, but you'll see there is a number of reviews, major articles, illustrative examples, different capabilities and limitations throughout what I'm presenting. And so hopefully it'll be a resource for you if you're interested in any of these different areas. Now, this isn't really a talk about different types of laboratory techniques that are pretty standard: transfer, dilution, extraction, reaction with pipetting systems and robots. It's about these droplet on-demand dispensers themselves and the various types of technologies. And I'd like to say there's not many of these droplet on-demand technologies made explicitly for mass spectrometry. So what I put together are some obvious analytical advancements in this area and trends related to these droplet on-demand dispensers for mass spectrometry. Today, because of limited time, I'm only going to talk about the droplet on-demand dispensers used with an open port sampling interface coupled with mass spectrometry. For those of you that are more interested in these other details that I'll overview, you can go to Shivo and you'll see a link at the end, and they have the full presentation with all of those details available for those that are interested. So non-contact droplet dispensers, they have various modes of operation and different droplet metrics. So can you hear me there? Did I unmute myself?
Yes, we can hear you.
Okay, sorry about that. Something popped up that told me I wasn't. Bulk loaded or continuous flow systems, mechanisms of dispense, pneumatic, electrostatic, piezoelectric, inkjet, or acoustic. The volumes and range can vary among these different types of dispensers, the rate at which they can dispense, the orientation with which they dispense, vertical up or vertical down, or all orientations. Now, for those that are interested, here's a number of review articles or overview articles that cover this area. So the first two here are general device applications, and you'll see that these droplet on-demand dispensers are kind of getting a new life in the bioprinting area. So you'll see a lot of them discussed when you see bioprinting. And then I've arranged here different types of different reviews from kind of large droplet dispensers down to smaller droplet dispensers, all the way down to scanning probe microscopes. So these are good references if you're interested in this area to go back and read one or all of these articles. So droplet sizes, what are we talking about here? So droplet volume goes with a cube of radius. So if we have a typical kind of droplet from a normal pipetter, a microliter volume, we have a millimeter sized droplet. If you reduce that volume by 1,000 times, the diameter reduced by a factor of 10, and so on. And so if you get down to attoliter sized droplets, the only way currently available to make those is with things like scanning probe microscope probes. But then from femtoliter all the way up to nanoliter, there's different types: electrostatic, piezoelectric, pneumatic, acoustic, as I've talked about. And what I'm interested in here for the mass spectrometry is this picoliter to nanoliter volume range. That's what I'm going to discuss. So here's this overall view of where these droplet dispensers can be used in mass spectrometry. And I'm only going to look at one subset today. But in sample preparation for things like matrix-assisted laser absorption ionization, you have things that are used for liquid chromatography, MALDI coupling, putting down the matrix, putting down the sample itself, derivatizing the sample, or making very small wells on a surface and doing reactions and other chemistries in very small wells for further analysis, or actually putting droplets on surfaces so you can do extractions from spatially resolved surface droplets. And then what I will focus on today is this opening pore sampling interface is a new type of interface for coupling to a mass spectrometer. But you can also, there's also in the literature continuous flow paper spray and other types of surface extraction methods coupling both the sample transport to the mass spectrometer and ionization. And of course, there are droplet dispensers that have been used for direct ionization, droplet electrospray ionization sources, droplets to different types of probes that emit ions, and droplets to inductively coupled plasma mass spectrometry, which has been used for a couple of decades for either single cell or different metal particulate type analysis in that area. Like I said, I'm going to talk today just about droplet dispensers with this open port sampling interface and some new technology that's available that takes advantage of that. So what are these open port sampling interfaces? So this is something that I worked on since the early 2000s. If you go into the literature, you'll see names like surface sampling probe, liquid microjunction surface sampling probe, open port interface, liquid vortex capture probe, touch express, and maybe even more names. These are all variations of different techniques that we came up with while we were at Oak Ridge National Laboratories. Many of these patented, many papers published on them. But in general, this family of interfaces are continuous flow coaxial tube devices. So two tubes aligned with an annulus that delivers the liquid. The inner tube delivers the liquid also to the mass spectrometer. So the tube size, their positioning, dimensions can all be optimized for particular applications. They can be used in contact sampling or non-contact sampling, and coupled with any kind of atmospheric pressure ionization source used today with mass spectrometry, whether it's electrospray, atmospheric pressure chemical ionization, atmospheric pressure photoionization, inductively coupled plasma, and so on. And you can configure these types of devices to collect the sample in like a fraction collector, or it can be connected directly online with the mass spectrometer. So what I want to talk about today is actually using one of these devices to capture small liquid droplets and the advantages that can be gained with that. So here's just like a Model 1 setup, these two coaxial tubes. You can operate the system such that it overflows, so there can be a capture well to capture that overflow material. Here shows, there's a video on the right showing these two coaxial tubes, the outer tube and inner tube. The annulus region between the two is where a liquid is pumped in. The Venturi effect of a pneumatically assisted electrospray, or APCI, is used to pump that liquid through this inner tube into the ionization source. So it's operating now in what we'd call a vortex mode, so it's sucking some air as well as the liquid. But you can change that flow rate and actually overflow the system. Liquid's still going to the mass spectrometer, but now the system overflows and washes itself. So this is a mode, for example, if you're going to touch a sample to the top, it's continuously cleaning. The vortex, or there are some other modes, subcritical vortex mode, are used for real fast sample transport to the detection system. So we've done a number of studies with microliter sized droplets as well as nanoliter sized droplets, and it's the nanoliter sized droplets I'm going to talk about. There are a number of different types of dispensers that could be used for this area, They can be robotically controlled. They can be in microtiter plates, which is very useful infrastructure. And you can use very small volumes in those wells, a few microliters. You only consume nanoliter volumes, and you can get very high throughput. So here's one example. This is a commercial instrument from now owned by CellLink, but this is Dispendex, their eye drop system. This is used a 96 well plate. I think they use 384 as well now. Can be used with different analytes to create arrayed material on a capture plate. And so we use this type of device with our open port system to actually read out microtiter plate system. So this is that microtiter plate in the IDOT system, a pneumatically injected droplet from a small hole in each of the wells is directed into one of these open port interfaces that's connected to the mass spectrometer. And here you can see that sample well plate. And instead of a capture plate where you can deposit the material wherever you want, we have this open port that we're directing underneath the well where we want to sample. And so this is just one particular result. So we're using this particular analyte at nanomolar concentrations. We're dispensing 5 nanoliter droplets, one at a time. In each case, we're looking at 12 repetitions. So we're in this vortex mode of operation. And you can see we're varying the time between the droplet dispense. So the droplet's coming out of the same well. Here it's a 10-second pause between each 5 second, 2 seconds, and 1. And so you'll see we can analyze without any kind of peak overlap about every 2.5 seconds. Now we can dispense at a much faster rate with these types of devices, but this particular throughput's limited by the design of this particular open port interface. That's not a fundamental limit. How you design it determines the kind of throughput you can achieve. But you'll see relatively low sample amounts dispensed, very nice signal to noise, and you can go from a single peak to a continuous infusion mode. So if you had a new sample you wanted to analyze, for example, optimize the mass spectrometer, you can get a continuous infusion mode, optimize the system, but only use very small volumes of the sample to achieve that. This is just an example showing that you can use the system well to well. So 2 different analytes, one in well 1, one in well 2, shifting back and forth, dispensing from the separate wells. And here showing 5 seconds well to well is easily achievable, baseline resolve. Again, about 2.5 seconds would be the type of throughput sample to sample you could achieve with this system, with a particular open port sampling interface that is used, but relatively high throughput using only nanoliter volumes of sample, great signal to noise. Here's another system that's very similar. This is the Satana single cell printer. This is also part of CellLink. And so now you've got a picoliter volume dispensing system. And you also have an optical system that allows you to look at cells that are being dispensed. And so you know when a cell is dispensed, you have an optical image of it, you can identify it, and then for each cell that's dispensed, you have a mass spec signal to go with it. So this is very useful for rapid cell analysis, single cell analysis. Now this is a new commercial device from SciEx, and this is based on an acoustic dispense system. The commercial version is based on the lab site, and so you'll know the lab site from their Echo system. This is called the Echo MS from SciEx. Acoustic dispensing system, 2.5 nanoliter droplets is normally what are dispensed. Our open port system is normally vertically upright, but for this system, this commercial system, it's turned upside down. It operates just fine that way. Individual droplets are injected from well plates into this open port and then carried on into the mass spectrometer. So you can have 384 or larger density well plates with this system. So an acoustic droplet ejection from each well plate, and that material into the open port is diluted during its transport to the ionization source. This dilution can take care of many of the matrix effects that one won't normally have in a mass spectrometry experiment that require lots of front-end cleanup. So this is kind of a very sophisticated or simple, depending on how you look at it, dilute and shoot type approach with mass spectrometry. Again, you're using an electrospray ionization source, pneumatically assisted for ionization. So we know electrospray very well, so that's a very stable and understood source. There's no fundamental reason that this could not be atmospheric pressure chemical ionization or atmospheric pressure photoionization. The current commercial version, the vendors chose to sell it with electrospray only. Now, here are just some of the results that can be obtained. And so this is a different design of the open port sampling interface, so you can get a little bit better throughput. So this is 1 Hertz sampling, or 1 second per sample dispensed from the same or different wells, very reproducible. A nice linear dynamic range. The detection limits with this system are in the low nanomolar range. Very little carryover. The system's continuously flushing itself. The volume of the droplet's very small, so actually the droplet's much smaller than the diameter of the inner tube. So there's very little of the sample ever hits a surface in this system, so very difficult to contaminate it. You can extend dynamic range by rapidly dispensing more than 1 droplet, so the system's basically seeing a larger volume. So from 2.5 nanoliters up to 25 nanoliters can be injected to increase your signal levels. Again, you can look at single droplets, or you can rapidly dispense so that you get a continuous infusion. Again, this is useful to tune the instrument or do things like extended tandem mass spectrometry experiments that take some time. But again, consuming very small volumes of sample, only 2.5 nanoliters per droplet. So a minute of this type of work consumes less than a microliter of sample. You're not limited to small molecules. This is electrospray system, so anything you can electrospray, you can use ionize with this system from small molecules, and here is showing antibodies, so proteins can be done. And again, this dilution effect that's built into the system, if you look at 2 different analytes from higher to lower concentration with and without a matrix that normally suppresses the signal, you see that you overcome that matrix effect. There's very little matrix suppression because of that online dilution approach. So if you look in the literature, there's a number of papers in the last 6 to 12 months. Most of these are from the big pharma looking at high throughput assays. SciEx has been involved in most of this work, and so again, I'll say at the end, people that are really interested, Tom Covey at SciEx would be a person to talk to about this instrumentation in detail from the research aspect. But here shows a number of different types of things that can be done with this system. So that commercial system showed you that you could do about 1 sample per second. And there are some samples, or actually papers here, that show that you can do even higher. Here's 6 samples per second with this system. But in the research lab, yet we have other systems that are even faster. So this is a case where we're making a very short open port sampling interface. It's only 4.5 centimeters long. This can be built right into the ionization source of the system. And for proof of principle, we're using this 3D dispense Nanoliter Droplet Dispenser. The current commercial version of this is available from Shivo. It's called the Nanoliter Dispense Module, or NLDM. It's a pneumatic dispenser, single nanoliter volume droplets at rates up to 20 Hertz. And so this is being used in the research lab to show that, in fact, you can do a much higher throughput with this system. So if you look here, we're dispensing at 10 Hertz out of this system. The baseline width of these individual pieces is about 68 milliseconds. That translates into the possibility for a 15 Hertz sampling rate, or 15 samples per second. So now this is pushing the ability of the mass spectrometer to detect even things like single reaction monitoring mode, because of the time you have to dwell to get sufficient signal and the number of points you want to cross the peak. So this can be, and we expect this to be even faster in the future, a very high throughput mass spectrometry approach. And then just to say a little bit more, if you're interested in this type of dispenser, Shivo has this on their website. Rich Parker, who's also listed here, is the person to talk to there. This is very nice because it's simple to set up these experimental systems, but it can also be ganged together, be used with commercial robotic systems to build systems for whatever you might need, whether that be building arrays or different research projects like I'm talking about here. So what you'd find in that full presentation that I said again is available from Shivo is the fact that there are a number of different types of discrete droplet dispensers that are out in the world. Several of them are made for mass spectrometry, such as this CHIP 1000 system from Shimadzu, or their LCMS LC MALDI fraction collector system. And then there are a number of others that people have put together, whether they be piezoelectric spotters or others, and the Shivo system here. But then the one real commercial system right now is this SciX Echo MS, which uses the acoustic droplet dispensing. I put together here a table to kind of show the merits and costs of these particular systems for those of you that are interested and might have use of these in other experiments, and then of course the details for that commercial SciX Echo MS system. With that, I'm going to stop, hopefully answer some questions, and again I point you to the people at SciX listed here, or people at Shivo for information on those two commercial systems that I emphasized in this presentation. Thanks again.
If you have questions, please open the chat and then send your questions to all panelists. I have a question, and it revolves around the, let's just say, carryover in the mass spec with baseline resolution. You were indicating in the beginning, I think there was a 2.5 second space between baseline resolution, but then you have examples with the nanoliter dispensing from Shivo. What is the next frontier of technologies that need to be addressed to increase the sampling rates? Is it, on the dispensing side, or is it on the mass spec analysis side?
Yeah, very, very good question. So I can't speak for the mass spec companies per se, but when we're up to about 15 hertz now, we are really pushing the ability for the mass spectrometer to detect a single analyte and certainly multiple analytes at once. So 5 millisecond dwells are kind of like low end right now minimums. So if your peak width is only 60 milliseconds wide, that's about 10 or so points across the peak for a single analyte. So that's a real limit there. It's a good limit to have. We didn't have that before. That's much faster than any other kind of high throughput mass spectrometry system. And then from a practical standpoint, when we're using something like the Shivo nanoliter dispense module, we're looking at the same sample over and over and over. So if we've got something like the Echo MS system that uses the lab site acoustic dispense system, or something like the IDOT system, well to well, those systems can only move at about 300 milliseconds. So we can't match a dispenser with the throughput at this point. So there has to be some real advancement in these dispensing systems for things like their well plate dispensers to take advantage of the achievable throughput with the open port.
So that'd be a limitation on the motor technologies actually physically moving between samples?
It's not so much, that is definitely one thing. You need some really good rails and repeatability in those. But if you do things like the acoustic droplet dispense, and you do it with some analytical control. So if you don't put the same volume in every well and you don't have exactly the same solution, using the same acoustic dispense parameters can be problematic in generating the same types of droplets. So there are some, they call them pings, but they use acoustic methods to actually figure out how much solvent's there and what type of solvents are there, so that they know what energy to apply to get the droplets. So if you don't know what's coming, you have to use those techniques, and that slows you down a bit. Now, it slows you down by tens to hundreds of milliseconds. It doesn't slow you down a lot. But if you say I've got the same wells, everything's exactly the same, then you can avoid them. But that's never going to be the case in a real analytical situation.
I also have another question about analyzing cells. So it's probably a naive question, but is there any fouling that you can expect when you're analyzing a system that contains cells?
Well, you're looking at one cell at a time, and they're, you know, it's a very small volume of material, one cell at a time. So certainly the system itself that's dispensing, you've got to get your concentrations right so that, you know, you're able to get one cell to dispense at a time. But I would say that, you know, probably thousands, tens of thousands of samples before there's going to be an issue.
And I guess the last question is, do you foresee any future applications or developments for droplet dispensers? You kind of went a little bit into that earlier, but anything on the horizon that you can forecast?
Again, I would point people to my full talk that's available, I think, on request from Shivo. But so there are people that are doing a lot of gas phase chemistries right now with single droplets, and so putting the droplets into those devices, and then those devices have to dispense to the mass spectrometer some way. So that's going to be one area. And I don't think things like matrix-assisted laser absorption are going to go away. One thing that's very big now, I think Dick Smith's group at P&L and others related to him are doing these techniques called nanopots, which are taking advantage of very tiny reaction wells to do chemistries, whether it be in proteomics or other areas. And so using these droplet dispensers to feed those wells or fill those wells is one thing. Then there has to be a way to read out those wells. Using conventional pipetters and syringes is an issue. So if those wells could be not only filled by these dispensers, but those wells were also dispensers themselves, that would be I think a really nice use for, you know, a new type of droplet dispensing technology.
Well, thank you very kindly. This is an excellent overview. I think we'll turn it over at this time to Rodney in closing statements.
Yes, thank you very much, Gary, and to Rick for fine presentations today. Thanks to Ron for asking the questions. We'd like you to know that we will post each of these two rapid fire presentations on the LRIG YouTube channel, and we'll send out an email to all registered attendees to let them know when that's up. So if you need to get some more information, I'd encourage you that if you would like to make a presentation at a future meeting, or you would like to organize or have an idea of someone who would be good to present, please send your proposal or ideas to talks.phl@lrig.org. That's talks.phl@lrig.org. Thank you very much and have a nice day. Take care. Bye.